phylogenetic trees and hypothesize about biology is you can use about different types of data. Molecular data is one of those things, and it's a technique and a tool we didn't have in the past, but it's become very useful nowadays because many times there are advantages to using this type of data, taking away some of the subjectiveness and the interpretation, factoring out that environmental effect on phenotype. And in many cases, the phylogenetic trees we get from this type of data match what we have for morphological data, so that's extra support for our hypothesis. We probably haven't seen much of this before unless you talked a little about some of this in Biology 140. My guess is a lot of this is new to most of us, and we may never have done these techniques before. But one of the first things that we have to make sure we can do is if we're going to use molecular data to construct phylogenetic trees, we have to determine what the sequence is of the nucleotides that is in that DNA strand that we have. Okay, so we're looking for what order are these nucleotides in? Guanine, cytosine, thymine, adenine, how are they attached together? What is that sequence? And to do that, we've got to make sure we've got enough DNA to work with, because most of the tests that we use to figure out what the sequence is destroy the DNA in the process. So if you've only got a little bit that you've taken from, for example, a fossil, an extinct species, that's not enough for you to run multiple tests or the number of tests you need to, and still have some left over in case you want to do tests later on. And so we talked about this new tool that has come into being within the last few decades, that we've really refined, at least in the last few decades. It's called the polymerase chain reaction, or PCR, that we can use to copy the DNA. Okay, very important. Polymerase chain reaction does not determine the sequence of the nucleotides. We don't know that. All we're doing with this process is we're just copying DNA. But I'm starting with this because the one method that I'm going to talk about that does determine the sequence is going to match a bit with this process. It's going to be very similar. So let's talk about what the steps are for the polymerase chain reaction. We just started in the first two steps in our last class. Okay, first step is you're going to be making a solution. That is essentially a combination of all the ingredients you need to do this process. One of the most important things to put in there is the DNA that you're interested in copying. That's your original DNA. It doesn't have to be the whole genome. It could be just a fragment. But what is it that you're interested in looking at and comparing between different species and looking for similarities and differences? What is it you're copying? Put that in. Put in something called primers, which I'm going to define in just a few minutes as to what that is. You're going to have an enzyme in there, some sort of a DNA polymerase, which is the enzyme that's essentially going to replicate and use the template you have there to actually add nucleotides and copy that DNA. Usually the DNA polymerase we use is something called Taq polymerase, and I'm going to explain why we use that one in just a second. And then you need a bunch of free nucleotides, just free adenines and guanines and thymines, all those kind of floating around in the solution, because that's ultimately what you're going to draw on to build your new chain, okay, your new DNA strand. Now, if the plant was doing this in nature, or us for that matter, because we do copy our DNA, our cells copy our DNA, we would undergo a process that first takes this DNA strand right here and unzips it. Because we're going to use both halves of that DNA, because we know DNA is double-stranded, both halves of those DNA as a template, okay, to copy. You can add nucleotides to this one to make it a new double-stranded DNA strand. You can add nucleotides to this one to make it a double-stranded DNA. So you're going to take one strand here and create two from that. In nature, the way we unzip that DNA is we use an enzyme to do that. But we're going to use an artificial process to do this in the polymerase chain reaction. Instead of putting an enzyme in there that unzips the DNA, we use heat instead. Okay, so if you know anything back from Biology 140, you probably talked about a word called denaturize. Most likely you talked about it in association of proteins, not necessarily DNA, but DNA is a biomolecule. It is capable of denaturizing as well. It's basically unzipping or coming apart is what that means. And when you apply heat to a lot of biomolecules like this, that's exactly what they do. Proteins tend to unfold, which means they don't function properly, and DNA tends to unzip. Okay? Now you might think, well, wait a minute now. We've also in this solution here got an enzyme that we've put in. And you would think if the DNA, it's hot enough for the DNA to denaturize, the enzyme would denaturize as well. But that's actually not going to be the case. It used to be the case. We'll talk about that in just a little bit. In the first DNA polymerases they were using, but now it's not because of this new one that we tend to use, and I'll explain why it's a little bit resistant to this heat. Okay? But we're unzipping the DNA so that we have basically two templates to work with. Each one of those is going to become a complete strand of DNA once we're finished with this. The next step is that your primers that you put into your solution are going to bind to your denatured DNA. Now, what are primers? Primers are small pieces of single-stranded DNA that you make, right? That the researcher makes themselves, that are designed to match specific parts of the sequence of the DNA that you're copying at either end of interest. So you notice we've got one primer right here that binded to this end of the top portion of the DNA strand. We have another one that binded right here. Okay. Now you might ask yourself, wait a minute, if I'm making this, and I've got to know ahead of time what this sequence is that it's going to bind to, how do you do that? If you have a DNA strand that you've never seen before and you have no idea what the sequence is. And there's a couple different ways you can do that. One, you can look at something that's closely related that you do know the sequence for, and assume that there might be some similarities in the DNA sequences between the two and use that as potential primers for the one you're copying. Two, you can also cut that DNA and put something in that you already know, right? In which case you can have the primers bind there and it will extend that into parts that you don't know, right? There's a couple of different ways you can do this. But you need those little primers to get you started, right? So it's going to attach there first, and this is essentially where the copying is going to begin, is right at that point. Next thing that has to happen is your DNA polymerase, your enzyme, now has to start the process of copying this. It binds here to the DNA, right where that primer ended, right here where the primer ended, and it starts grabbing all these free nucleotides that are in the solution, the correct one, and putting it where it's supposed to be according to the code that's on this one half of the DNA. It just keeps adding it, adding it, adding it, adding it, adding it, right, until it's finished. Right, and then by the time you finish this step, whereas before you had one DNA strand, you now have two copies of the same thing. Okay? But that's not enough. You need more than that. Right? 2 copies is not going to be enough for the procedures that you want to do for the sequencing steps. So you want to keep doing this process. You do it over and over again. The cycle continues over and over again. So the next step then would be to heat it again. And when you heat it, you're unzipping now both of those strands. Okay, so you're getting 4 templates now instead of 2. The primers are going to attach to those. Right, we've still got primers in here. The DNA polymerase, they're going to start copying both of those, and you're going to end up with 4. Then you'll heat it again. Those will denaturize. And those 4 will become 8. And then you do it again. Those 8 will become 16, and so on and so forth. So it grows exponentially. And you just keep doing that until you get enough DNA to work with for the process. Again, this is just copying it. Okay? The next step after you have enough DNA to work with is now to do another procedure, a different process, to figure out exactly what these sequences are, what the sequence of the nucleotides is. And there's a lot of different ways you can do that. Okay? Now, going back to why we're using this particular enzyme as our DNA polymerase for the DNA process. This thing's called Taq polymerase. It is a type of DNA polymerase. Its job is to add those free nucleotides to the template as it copies the DNA. Initially, right, this process was pretty inefficient. And the reason for that is because of that second step in the process that involves heating up your solution to fairly high temperatures. It requires the use of some sort of an enzyme, right, that can withstand temperatures that are greater than 90 degrees Celsius. That's pretty warm for a protein. Most proteins are going to denaturize if you warm them up to that temperature. But that's how high you have to get it in order for the DNA to unzip, to denaturize. But you don't want your enzyme to denaturize at the same time. Right, so we've got to have something that can handle those temperatures that are above 90 degrees Celsius. Okay, well, initially, the DNA polymerases that we used couldn't do this. And so what we ended up having to do is we ended up having to put really, really large quantities of that enzyme in there to make sure that at least some of them survived this heating process and were actually able to copy the DNA. And we had to have continuous handling throughout the process, in some cases continually adding those DNA polymerases. Not very efficient. It was very time consuming and it was very expensive. It was very clear that they needed something much more efficient or a better way of doing this, if they could find it. And so the search went on as to whether or not they could find some sort of an enzyme that can handle this process and the temperatures that are involved with it. And they were able to find it in an organism that actually lives in hot springs. It was a type of bacteria that naturally lives in hot springs. It was a thermophile. That means it's an organism that can live and thrive, not just live, but thrive in hot temperatures. And that bacterium was called Thermus aquaticus, and hence the name Taq polymerase. The DNA polymerase that they took from this bacteria, they took the first letter of the genus name, the first two letters of the specific epithet, to spell TAC, to be that TAC polymerase, so that we know exactly what organism it's coming from. This bacterium lives in hot springs that are somewhere around 50 to 80 degrees Celsius. Right? And so the enzyme that it uses to copy its own DNA naturally is able to withstand and be stable at high temperatures, right? Even when the DNA may denaturize during the process of PCR. So this, just finding this enzyme, paved the way to dramatic improvements in the way that we do the PCR process. It was less expensive, less time consuming, less handling involved, and you didn't have to have as much of the enzyme that you added to your solutions. So kind of an important scientific discovery. And you notice it wasn't that long ago that we found this. Not that long ago, it was before I was born, but not that long before I was born, so not that long ago, because I'm not old. All right, so how now are we going to determine our DNA sequence? We copied our DNA, now we're gonna actually figure out our nucleotide order. But one of the methods that you can use for this is something called chain termination sequencing. I'm using this one, and this is the only one I'm talking about because it is so similar to the PCR process. Essentially, everything's gonna be the same, except for you're going to add one additional thing to your solution that you didn't add before, okay? So think back again to what you add for the PCR process. You have your original DNA that you want to copy, right? You have primers that you're adding, you've got to have those too. You have your enzyme, your DNA polymerase, we're adding that one, right? And then you had free nucleotides, right, that we're going to be pulled upon to build the new strand that you're creating with this copying process. The additional thing that you add during this chain termination sequencing process that's actually going to let you figure out exactly what order the nucleotides are is you add special nucleotides that are called terminator nucleotides. I know that's a really cool name, right? Which I've always called that. Terminator nucleotides that have special fluorescent dyes attached to them. And each one of the different nucleotides has a different color. So you'll have a certain fluorescent dye that you use for adenines, right? For these terminator nucleotides that are adenines. You have a special color for the cytosines, you have a special color for the thymines, and a special color for the guanines. And then once you get those put into there, you start the same essential process that you did before. You heat up the solution, let the DNA denaturize, and unzip, have the primers attach, the DNA polymerase starts adding on nucleotides, but remember, you don't have just normal nucleotides in there, you also have these terminator nucleotides in there as well, right? So it's going along, that enzyme's going along, and it's adding on normal nucleotides, and all of a sudden, at some point, it's gonna grab one of those special ones that has the fluorescent dyes, and it's gonna add that. And because of the chemistry of these terminator nucleotides, that's as far as it can go. It stops the copying process at that point. Now, it can grab a terminator nucleotide at any point in time, right? At any location it wants to. Just whatever happens to happen and grab it, that's when it stops. So what you're gonna end up with is you're gonna end up with all these fragments of DNA that are all different sizes, depending on when the terminator nucleotide was added in the process. So here you can see they've got an example here. We've got one that's got just one base pair on it. This one's got 7 base pairs. Before it actually added the 7th one was when the terminator nucleotide got added. We have 4 here, 1, 2, 3, 4, 5, 6 here. This one has 2, 1, 2, 3, 4, 5, 6, 7, 8. We've got an 8 in that one, 3, and so on and so forth. Right? So we've got all these different length fragments now. What we're going to do next is we're going to undergo a process called electrophoresis. Electrophoresis, okay? What this is going to do is you're going to take these fragments that you just formed and you're going to put them into a gel. And you're going to subject that gel to a current that's going to have a positive charge on one side of the gel and a negative charge on the other side of the gel. Now, if you remember anything about your chemistry of DNA molecules, you'll remember that they have phosphate groups attached to them. And those phosphate groups are negatively charged. Negative is attracted to which charge? Positive, exactly right. Right, so here's our negative side. That's where we put them in. That's where we put the fragments in. We run the current through this, and then the positive side of the gel is over here. That's what the DNA is going to be attracted to. So it's actually going to start to migrate through this gel. But it migrates according to its size. The smaller the fragment, the faster it moves, which means the further it goes. So small fragments are going to move very quickly, and they're going to go very far in the gel. So here you can see, because this one's only one base pair in length, it was the one that moved the furthest in the gel. This one had 2, so it moved the second furthest. This one had 3, so it moved the third furthest, and so on and so forth, right? And you'll have some sort of standard ladder there that will help you figure out exactly what size these fragments are. And since you know what size they are based on how far they move, and you know what the very last one is because of the color that they shine with that fluorescent dye, you know what nucleotide is in that position, okay? So in position 1, we have this. In position 2, we have this one. In position 3, we have this one. Position 4, we have that one, and so on and so forth. So you can actually come up with what your sequence is supposed to be. Yes, it's time consuming, but it works. It takes time, but it's a very effective method of doing this. Right? It's not even one of the most modernized versions of doing this, but it is one that's still used frequently today. And the only difference in the process is what these special terminator nucleotides are that you're adding to the solution. Everybody feel comfortable with this one? Yes?
So they're basically shocking it with electricity to separate it so then they can determine the sequence.
I wouldn't say shock, because it's not a matter of we're not doing that. You actually have a constant current running through it, and it's not necessarily a really, really high one, because you don't want to destroy the DNA, obviously. But it's enough that you've got the 2 charges there on 2 sides of the gel that cause those fragments to move and be attracted to the positive side.
But they're doing that just so they can determine the sequence?
They're doing it to make the DNA move. And they want it to move, and they want it to move in proportion to its size. And this only works if you have these different size fragments. Anything that's the same size is going to move the same distance, but if they're different sizes, they're going to move different distances, and that tells you how many base pairs there are before that terminator nucleotide was added. And since you know that's the last one, you know what position that's in then. We good on that one? I know this is new stuff, right? Okay. Well, now we have our sequence information. So now we need to use this to come up with a phylogenetic tree. So how do we take this data, and how do we use this to construct our hypothesis for our phylogeny? It's kind of the same way that we did before, right? If you recall, in the past, the very first chapter or PowerPoint that I taught you, we were talking about describing organisms, and we said that you assign what are called characters to them. And you look at the character states that exist for each of the different species. So flower color, for example. Every angiosperm has a flower color because they all have flowers. The different character states might be pink or blue or purple or white, depending on what the species is, as to what their character states can be. You can do that same concept here with molecular data. Okay? So what we're going to do in this case is we're going to take the DNA that we have from different taxa. Let's say these are different species. So we've got species 1, species 2, species 3, species 4, species 5, species 6. Okay? And we're going to take those DNA strands, right, and we're going to line them up with each other. Right? That means that you can look here, we actually have different positions, right, all along the DNA strand here that we can number based on where we are in the DNA sequence. This is position number 1, this is position number 2, position number 3, position number 4. And we can compare those from one species to the next. What is at position 1 for species 1? Right? What is at position 2 for species 2? Right? Is it a G? Is it a T? Is it an A? Is it a C? Right? And that's exactly what they're doing here. They're using each one of those positions as a character. That's why we get so much data with molecular DNA when we look at the molecular genetics, is because every single one of these positions are things that we can compare between species. That's a lot of data that you can compare, more than morphological data. So our character in this particular case is our nucleotide position. What number position are we at right here? Right? And then our character state is going to be whatever nucleotide we have at that position. There's only 4 possible choices. Do you have an adenine there, a cytosine there, a guanine there, a thymine there? Okay? So go to each of the positions and do that. They did this where they actually number coded each of the different nucleotides. And I'm only pulling out some of the character coding information here. I'm specifically pulling out what they saw at position number 1, what they found at position number 2, and so on and so forth. And you can see it color coded to kind of help you which positions I'm looking at right here. Okay? So for example, they gave anything that was an A, a 0. Right? So if adenine was in that spot, they put a 0 down, right, in that one. If they had, instead of an A, they had a C there, they gave cytosine number 1 as part of their code. For numbers 2, that would be guanine, okay? And if they have a 3, it means it was a thymine, right? So we go through it in our position right here for taxon number 1, we had a G. They coded that with a 2, so they put a 2 down there for species 1. They put a 2 for everything, because everything had guanine except for this one down here, which had C. That's why they put a 1 down there in that red column. We go to this position here. We had A's here for species 1 and 2, so they gave those, the A has a 0. They had thymines for these two, so they put 3's down for those two, and they put 0's down for these two right here, right? And so on and so forth. So we're looking for similarities here, right? How many similarities exist? The more similarities that you have, the assumption is the more closely related you are, right? And you'd be closer together on your phylogenetic tree. Right? So that's what we do with this. We have an assumption. More similarities means the more closely related you are. Closer you would be on the phylogenetic tree. This applies to both morphological data and it applies to molecular data. You can have similarities in appearance and anatomy between organisms and construct the tree based on their morphology and have them closer together based on that, those similarities in anatomy. Right? But the assumption is if they have similarities in morphological characteristics, it's because they have similarities in DNA, right? That their molecular data is also similar. So here's an example. Okay. We've got 3 different species that we're comparing to each other in terms of the molecular data. We're going through, we're looking for similarities. You can see all of them are similar here in position number 1. When we go over to this one, species 1 and 2 are similar, but species 3 is different. Over here again, species 1 and 2 are similar, species 3 is different. And here, species 1 and 2 are similar, species 3 is different. And here, species 1 and 3 are similar, and species 2 is different. Right? But we have way more similarities between species 1 and 2 than we do between 1 and 3. That would then apply, or assume, that species 1 is more closely related to species 2 than it is to 3. Good assumption? Maybe, right? Here's the problem with this technique of making this assumption that the more similarities exist, the more closely related you are. You can actually have similarities that exist, right, and not be closely related at all either. Mainly because you live in similar environments and have similar adaptations. So let me give you an example of this. We have 2 plants right down here, right? If we were going on this assumption right here that these two, the similarities that we have here between these 2 species would imply that they are related to each other because none of them have leaves, right? And they have very thick column-shaped stems that they store water in, right? Both of them do that. And they have spines. We might say, these have got to be closely related. We're going to put them together on a phylogenetic tree. That's actually not true for these plants. This one on the right, which is in the family Euphorbiaceae, the spurge family, is actually more closely related to this thing right here than it is to a cactus, despite how similar they look. Right, that's why I always tell students, I say, you're not going to find cactus growing in deserts outside of North America, unless they've been planted there, because the cactus family evolved in North America. That's the only place you find them. Right? You may find things in other deserts that look like them, but they're not cactus. They're not even in the same family, and they do not develop these characteristics the same way. They didn't have a common ancestor, right, that had this particular feature that got passed to all these descendants. So these are more closely related. These should be closer together on the phylogenetic tree than these should right here. Okay, so how do we determine that? Here's our problem. Similarities between species can be due to either shared ancestry or something called convergent evolution. My hope is that you were introduced to convergent evolution in Biology 130, and if you've had ecology or evolution Beyond that, you probably talked about that as well. But convergent evolution is where you have similarities that occur between distantly related species that have evolved them at separate times, right, in separate events, and in some cases separate places. Whereas something called homology is similarities that are due to common ancestry. In other words, some evolutionary trait appeared in evolutionary history that occurred in this ancestor, and that feature, whatever it is, got passed on to everything that is descended from it. Unless something happened later on that changed it, you would expect all of its descendants to have this feature because it was in the ancestor. That's what we refer to as homology. If instead you have this similarity that evolved separately, independently, in these two groups at different times, and their ancestor doesn't have this feature, that is called homoplasy. That's the opposite of homology. It occurs when natural selection essentially favors the same solution to the same problem in similar environments, similar climates. And it can definitely occur in distantly related species. As long as they're living in the same habitats, or the same type of environment, what is adaptive in this environment, whether it's in North America or Africa, is still going to be adaptive because they're under the similar conditions. And I'm going to give you an example for that. Those two plants that you just saw, the cactus and the euphora, the spurge, live in desert habitats. One of the most significant dangers that you have and risks and challenges that you have associated with desert habitats is a lack of water. So they both have these column shapes and three-dimensional stems that allow them to store lots of water. And they have spines to protect them from herbivores that may eat the plants to get water from as their source of food. Because there's no places to drink from a pool in a desert, most animals are going to get their water from what they eat, either the bodily fluids of the animals they eat or the plants that they eat. So a lot of things in deserts are spiny for that reason. Because it's going to be adaptive to be able to store water and have spines no matter what desert you're in. Doesn't matter what desert you're in. If you're in dry conditions, that's going to be adaptive. So how do you determine whether or not these features are a result of convergent evolution or homology? You have to look at how they develop, right? What the developmental path is for each of these. So if we specifically look at our cactus here and our spurge, and we're going to focus on the spines in this case. The spines for the cactus develop in a very different way than they do for the spurge. The cactus actually has its spines that are modified leaves. Those are the leaves of the plant. They don't really need the leaves to photosynthesize because they've evolved to have their stems do that for them. That's why they're so big and three-dimensional, right? But these right here are what the leaves are, right? So if you look at how they develop, what cells they come from, where the genes are to make those, you'd find it all related to the leaves. Whereas the one on the right here, these are modified stipules. Okay? Stipules, if you're in Tuesday lab, you know what those are already. If you're in Thursday lab, you haven't been introduced to those yet. These are two appendages that sometimes appear at the base of leaves, where leaves attach to the stem. There's always come in pairs because there's one on either side of the bottom of the leaf down there. And you can see that's why there's two of these here, as opposed to one, because this isn't the leaf, these are the stipules. And so they develop from different genes in a different way from different cells than what the spines of the cactus develop from. So we know this is a result of convergent evolution. These two species evolved this characteristic independently at different times, in different events, and in different, potentially different deserts. So it does complicate things a little bit. So how then do we use this information to make the phylogenetic tree? Because we know these are going to have to be separated. And we're going to have to talk about two different events, two different derived traits that appear on the separate lines going to those species to indicate that they evolved those separately and independently. Right? Well, what we're going to do here is we're going to look at all the potential possible ways that you can construct the phylogenetic tree, all the different ways you can branch it. And we're gonna use something called the principle of parsimony to decide which is the most likely pattern. There are other ways of doing this. This is one of the more common ones for making phylogenetic trees, is to use the principle of parsimony. Parsimony states that the most likely explanation, in this case, the most likely tree, that is correct, is the one that implies the least amount of change. And in this case, it is evolutionary change that we're talking about. The least number of evolutionary changes. And what that's going to imply is it's going to imply that homology should be more common than homoplasy. Because it's more likely that there is a single evolutionary event that occurs that leads to multiple species having that because they're descendants, than to have separate events happen that lead to the same trait. That's less likely to happen. Doesn't mean it can't happen, it's just less likely to happen. And I'll give you some examples here. And we're actually going to use molecular data to do it. Since we're dealing with our molecular systematics chapter here, we'll use molecular data to make our trees here. Okay, so let's say that we've gone into our ancestor here, and we've determined what the DNA sequence was for this particular ancestor, and we are now looking at the descendants for that ancestor and trying to figure out how they should be arranged in a phylogenetic tree based on their relationships. Right, so here you can see, I'm going to use just a very small sequence of DNA just to make it easier for us to visualize this. This is what that original DNA sequence looked like in the ancestor. These are what it looks like in all the descendants. Okay? So if we go up and look at these, compare, we can see for this first descendant lineage right here, there weren't any changes that happened, at least on this particular part of their DNA. This part of the DNA of the descendant is the same as it was for the ancestor. It doesn't mean other changes can happen just for this section, it didn't change. For our next descendant that you see branched off next, there was a change that occurred. We went from having an A at that position right there to having a C. So I've indicated that on the tree, that there was some sort of a mutation or some sort of an evolutionary event that led to this adenine right here changing to a cytosine, and it became this new descendant. Then we have another branch coming off here, another descendant from this, where there was a change that occurred at this position where the cytosine became a guanine. So I've indicated that there was a change that happened right there. Right? Then we have another descendant. This is the change that happened for that, so I've indicated that on the tree, and there's the change that happened for that one. Right? That's just one possible explanation, right? One possible way that we could branch these descendants together, saying that every time it's branched off, one descendant went one direction and everybody else went the other direction, right? But we could also put it this way. These are exactly the same lineages right here. All of these are the same descendants that I've got listed up here, got the same ancestor right down here. The only difference is how I branched them, how I positioned them. The same changes occurred that happened before. So this is this one right here, this is this one right here, we've got this one, and we've got this one. Right? They're all the same, they're just rearranged in a different way in the tree. Right? So what we could do in this case is we could look at the changes that occurred and say, oh, okay, we still have these two right here that had an adenine change to a cytosine, so we could say that changed happened right here and right here, therefore it led to a difference between these descendants and the ancestor. These both changed to guanine, okay? So we've got two indications here that that changed, going to those two descendants right there, right? With me so far, right? This is just two possible ways that this could work. There are many different phylogenetic trees we can make beyond this. So what do we do with this? We look at all of them, and you need to count the number of changes in the DNA sequence that happened for each one of the trees. How many different changes had to happen for this pattern to exist, right? What you're doing here is you're counting those tick marks. How many of these little tick marks do we have in the tree? Because it's indicating how many times a change had to happen. For this tree right here, how many changes occurred? Four changes, right? Right? 4 evolutionary changes for that tree. The second one? 4 as well, right? Okay, 4 evolutionary changes. They're just in a different position, a different place, right? But still 4 changes. Now so far, just comparing these two trees, both of them are equally likely to be the correct hypothesis, right, based on the principle of parsimony, because both of them involve the same amount of change, right? They both involve 4 changes. But can we get that number lower? And the answer is yes, we can. What I'm gonna do in this case is I'm gonna use this same exact tree that we have right here. I'm not gonna change anything about the branches or where the descendants are. The only thing that I'm gonna do is I'm gonna change the position of our derived traits and do it this way. Instead of having two changes happen here that are essentially the same change that lead to these two descendants, I'm gonna put it lower on the tree and say, no, that happened earlier in this ancestor down here, and it then got passed to both the branches that descended from that ancestor. Okay? And this one, since it's the same change, I've moved that one down here to indicate that the change happened earlier in evolutionary history than up here, right? And it got passed to both of those descendants. Right? It still works, right? We still get the same result, right? But in this particular case, how many changes were needed? Only two, right? Right, now, go back to what we just talked about. Convergent evolution or homoplasy versus homology. Which is which? Okay? This one is an example of what? Homology, right? Because you have similarities between species because of shared ancestry. Their ancestor had the trait, therefore they have it. It got passed to both of them. These are both examples of homology. Okay? These are similar because of their ancestor. These are similar because of their ancestor. What is this an example of? Homoplasy, exactly right, convergent evolution, because instead of having this because their ancestor had this, their ancestor didn't have that trait. Their ancestor had this one right here. The change happened after that, and it happened separately, independently, at two different times, along two different lines. That would be homoplasy. According to the principle of parsimony, this is less likely than this. You expect homology to be more common than homoplasy. So, if you're using the principle of parsimony, you would assume that this one is probably the best fit and the most likely hypothesis. Okay? The fewest evolutionary changes, most likely scenario. So this would be the one they would most likely accept if they're using the principle of parsimony to make their tree. So on the test, this is fairly easy to do. Okay? You're given a bunch of different cladograms, and I ask you which of them, and it's all based on the same lineages. If I ask you which one of best fits the principle of parsimony, all you have to do is go in and count those tick marks and find the one that has the lowest number. Whichever one has the lowest number has the fewest number of changes, and that would match the principle of parsimony the best. Okay, now, one other thing to keep in mind too. These phylogenetic trees that we're making, whether they're based on morphological data or molecular data, are purely hypotheses. We do not know if that is in fact how those particular organisms are related. Is it possible that those homoplasy events were actually the true scenario that happened? Absolutely. And we weren't there for the life of the species. We weren't watching it every second of its existence to know whether these changes happened at separate times or if they were in the common ancestor. We can only draw on the data we have and make assumptions and hypotheses based on what we know about anatomy, okay, and changes. So these are hypotheses. They can be supported or rejected by data. We never prove them. And that's why the more cladograms you can make for a group of organisms using many different types of data, if they all support that hypothesis, that provides even more strength to your hypothesis, but it's not proving it. Even if the morphological data and molecular data match, that doesn't prove that this is their true phylogeny. It just provides extra support for it. Okay, everybody's glad we're on molecular systematics now? Awesome. I told you it would be fairly short. Okay. All right, now what we're going to get into is we're going to start getting into the different groups of plants. And the way we're going to approach this systematically is to go through the phylogenetic trees and just keep getting more and more specialized into smaller and smaller groupings. So the first thing I'm going to introduce you to is a group or clade that's called green plants. It's actually going to include more than just plants. It's going to include some protists as well. But they call them green plants. I hate that word, that combination, because it's just kind of confusing since this gonna include some photosynthesis. But let's start at the biggest cladogram we can, the most inclusive cladogram we possibly can. And that's gonna be our tree of life. This is the tree of life. In other words, this is the phylogenetic tree for every living species in existence that we know of today. Right? It's obviously, I'm narrowing it down here to the domains. There are only 3 domains in the tree of life: bacteria, archaea, and eukarya. Hopefully you remember that from Biology 130. Both of these 2 domains right here, all the species of bacteria, all the species of archaea, are types of prokaryotes, which means that they have prokaryotic cells. There are some features you expect to be missing from prokaryotic cells that are going to be in eukaryotic cells. Here are the things to know about prokaryotes. They are all unicellular. Sometimes they like to group together into colonies, but they're still unicellular. They have a single chromosome in them. And it looks like a big mash of things, a big mash of DNA inside of a region called a nucleoid that's not at all enclosed by membrane. But if you stretched it out, kind of spread it out, it would actually be linked end to end in the shape of a ring. It's circular. Now this is called a nucleoid because it doesn't have a membrane around it like a nucleus does, which is what we find in the other group of organisms. So they lack a nucleus, and they also don't have organelles. Things like chloroplasts, mitochondria, Golgi apparatus, endoplasmic reticulus, all that's missing. The reason for that is that they tend to be significantly smaller than eukaryotes. Being smaller cells means that they can do most of the jobs that they need to do by themselves. The cell can do everything. When you start getting bigger than that, like you do in the eukaryotes, you have to start to compartmentalize those jobs and split them up into different parts of the cell because the cell can't do everything itself. It's just too big, and there's not enough surface area to volume to do that. So in the eukaryotes, this is what you have instead. You have organelles to help you do the jobs you need to do, like cellular respiration and photosynthesis, those sorts of things. You have a nucleus, which is bound by a membrane, where the DNA is going to be housed. You're going to have multiple chromosomes. How many you have will depend on what species you're talking about. But if you stretched them out, they're going to be linear, not circular. And you can have eukaryotes that are unicellular, like some of the types of Clonus would be, and you can have eukaryotes that are multicellular. Plants are what? Eukaryotes, exactly right. So plants are eukaryotes. So they're going to have these characteristics right here. And we're going to get into some of their specialized organelles in just a little bit. Okay, now not all eukaryotes are photosynthetic, but there are a number of different groups that are, and not all of them are plants. Some of these are types of protists. There aren't any animals that photosynthesize. That would be super cool, but they don't. But we do have some different types of protists that can do this in addition to plants. Because all of these are eukaryotes, okay, and they have organelles that have to be specialized in certain jobs within the cell itself doesn't do photosynthesis. Their specialized organelles do, and the one that does it is the chloroplast. So anything that's a eukaryote that does photosynthesis is going to have chloroplast. Doesn't mean they're structured the same way, we'll get into that, but they will have chloroplasts. Now if you are a bacteria, which is a prokaryote that does photosynthesis, you won't have these, okay? Things like cyanobacteria, they do the photosynthesis on their cell membrane. They do it, the cell does it itself. It doesn't have an organelle to help them do that. Just eukaryotes have these chloroplasts. So how then did eukaryotes get those? And why do eukaryotes have them and not prokaryotes? There's a theory that is put behind this particular idea of where these chloroplasts, and for that matter mitochondria, as to where they came from, and why eukaryotes have them. It's referred to as the endosymbiosis theory. Hopefully you have seen this before. Have you seen it? Raise your hand. Yay! Okay. If you're in my 130 class, I know you saw it. Okay. We're going to get more specific here than what I do for my 130 class. We're going to talk about primary and secondary symbiosis. And some company music too. Alright, so the endosymbiosis theory suggests that chloroplasts and mitochondria, two different types of organelles that do different things. Chloroplasts do photosynthesis, mitochondria do cellular respiration. In eukaryotes originated as free-living prokaryote cells, more specifically bacteria. And that they were engulfed by some sort of an ancestral eukaryotic cell. And for whatever reason, they were not digested. They actually formed a relationship between the cell that took them in. So we're going to talk about that and how this relates specifically to plant chloroplasts. Now there are different endosymbiotic events that occur in evolutionary history. We're going to talk first about something referred to as primary endosymbiosis, because it is the one that happened first. It was the very first event where this actually occurred. So in primary endosymbiosis, you have an ancestral eukaryotic cell that engulfs specifically a cyanobacteria, or an ancestors of cyanobacteria. This is a type of bacteria that can photosynthesize. Okay? It took it in, and it didn't get digested for whatever reason. Instead, it formed a mutualistic symbiotic relationship with the plant that it was inside of now. Hence the name endo. Endo means that there's something living inside of another thing. So we've got a cell inside of another cell now. And symbiosis means that they're forming a very close ecological interaction with each other. Right? But right now, at this stage right here, we still have just a cell inside of a cell. That's it. We don't have a chloroplast yet. This is still just a cyanobacteria. Right? There had to be some changes that happened in evolutionary history for that to become a chloroplast. In particular, you had to have evolution towards reduction. What that means is that the bacteria changed structurally and functionally. It retained some of its abilities, for example, its ability to replicate. It can replicate on its own time. It kept some of its own DNA, but some of the DNA did get moved into the cell's DNA. But it lost its ability to live independently. That's when it became a chloroplast. So if you try and take a chloroplast out of a eukaryotic cell and try and get it to live on its own, it can't do that anymore. It's not a cyanobacteria anymore. It needs the cell for its own survival. But it had to go through this evolutionary process to get to that point. Now, things to know about organisms that have chloroplasts that are based on primary endosymbiosis. One, I want you to know the origin of this chloroplast. It was a cyanobacteria. That is what got engulfed. Right? A cyanobacteria. Because it's going to be different for secondary endosymbiosis. And the second thing to know is that the chloroplast is going to have two membranes around it. This is related to this process of engulfment, okay? So here's our little cyanobacteria right here. It is a cell, right? Which means it does have a membrane. All cells have plasma membranes around them. There's a cell membrane around this cyanobacteria. Then you have your eukaryotic cell right here that's engulfing this. It also has a membrane around it, right? When it brings it in, these two sides right here are going to come together and close. This, and in the process, some of the membrane of this eukaryotic cell is going to bud off and wrap around that cyanobacteria. There's a process of phagocytosis. Okay? So now we have a cell inside here that has its own membrane plus a new membrane around it that came from the cell that just took it in. Hence the chloroplasts have two membranes because of this. Now the other ones that we're going to look at are going to have 3 or 4 membranes associated with them, because they are the result of something called secondary endosymbiosis. Okay? What's going to happen here is we now have a eukaryotic cell here with a chloroplast in it, right? Okay, this is either a green algae or a red algae. I'll just tell you that right off the bat. What's going to happen in secondary endosymbiosis is another cell that is now going to come into the picture, and it's going to take this whole thing in. Okay? So in secondary endosymbiosis, you have a photosynthetic protist, either a green algae or a red algae that was engulfed, and that whole thing became a chloroplast. Complicated, I know. But this is why there are structural differences between brown algae, red algae, green algae, and plant chloroplasts, is because of this. Okay? Alright, so we'll stop here for today. I'm going to get more into the secondary endosymbiosis in the next class.